All right, welcome everybody. We're going to get started in about 2 minutes once we get a critical mass in the room. Uh, Welcome everybody. Give you about 30 seconds. We've still got some more attendees coming in, so just want to give folks a minute to get started after perhaps their lunch breaks. All right, let's jump in here. Happy Wednesday, everybody. We've got a pretty jam-packed episode of When Things Go Wrong. See you all the last day of January, if I recall correctly. I'm David Valancourt, CEO and founder of the GMP Collective. This is our 17th episode of When Things Go Wrong. As a reminder to everybody, this recording, as all of them, will be available on our website, gmpcollective.com. We also post it on our YouTube channel, so if you're not able to stay for the whole time, if you've got some colleagues that you know are really bummed that they missed out, please forward this information to them and let them know they are all available on demand. Just a couple of housekeeping items before I turn it over to Bethany and kick us off. We have received a lot of great questions during the registration process. We have done our best to incorporate them into this webinar discussion with Jeff and Chris. But that said, if other ideas come up, use the Q&A. You can ask a question, you can ask a question anonymously, and we will do our best to get to them. If not, we will also work to follow up with you. Please bring your questions to keep us engaged. This is for you all. This is for the audience. This is educational, and we want to know that we're hitting the mark and helping you guys with your burning questions on this topic. So with that said, we've got now down to 57 minutes of jam-packed content, and I will toss it over to you, Bethany, to take us from here. Thank you.
Great. Welcome, everyone. I'm happy to continue to be your moderator of When Things Go Wrong, where we discuss various challenges and issues within the cannabis industry with the aim of offering valuable insight, tips, and solutions based on our guest experts' experiences. I recently joined the team at the GMP Collective, as many of you know, as Director of Content Strategy and Market Growth, and I am excited to continue working with these brilliant and passionate experts in the cannabis and hemp industry. So today's episode, let's get started. You all know David Valancourt, CEO of the GMP Collective, co-founder of the S3 Collective, and vice chair of ASTM International Committee D37 on Cannabis. We also have Chris Hudala joining us today. He's a PhD analytical chemist with more than 35 years of research experience in analytical chemistry focused on chromatographic and spectroscopic methodologies. He's the founder and chief scientific officer of Pro Verde Laboratories, Inc., a premier analytical research and testing facility for the regulated medical cannabis and hemp industries, and also serves on the cannabis expert panel with the United States Pharmacopoeia, USP. Also joining us today is Jeffrey Williams of Cayman Chemical. He's a synthetic organic chemist and manager of the phytocannabinoid research and development group at Cayman Chemical. His research group focuses on the synthesis and characterization of phytocannabinoid reference standards to support both cannabis testing labs and toxicology research areas. His group at Cayman Chemical has met the challenge of preparing standards for the many structurally complex compounds that occur naturally in cannabis, as well as those that result from chemical conversion of cannabidiol, their metabolites, and isotopically labeled analytical standards. So clearly we have some very knowledgeable folks in today. So let's go ahead and jump into the panel topics here. First, hemp is becoming a complicated, broad topic. Hemp is technically cannabis, but there's product you can get in a legally regulated dispensary versus a gas station or other store. There's marijuana versus hemp, THCa, delta-8, delta-9. And now we're getting into this numerical and alphabet soup, and it's getting a little confusing for somebody like myself. Let's get these definitions out of the way before we dive even deeper. Jeff, would you like to start?
Yes, I'd be happy to. Thank you, Bethany and David, for the introduction and the opportunity to participate in this forum. I'm going to try to offer some clarity on everything in this industry, but it's constantly changing. So we kind of have to put a warning label almost to say, this is what's happening on the 31st of January, 2024. There are constant changes that change our viewpoint on the issues around cannabis. So first, cannabis and its intoxicating effects have been known for millennia. It's only over the last few decades, or about half century, that we've known that the cannabis plant produces phytocannabinoids, which are responsible for the psychoactivity. And the biosynthetic pathways for those major phytocannabinoid constituents have been worked out. And so we know that cannabis sativa L produces these phytocannabinoid acids. And if we could have the first slide come up, the phytocannabinoid acids that the cannabis plant produces, the major ones are shown here in blue, and they're cannabigerolic acid, or CBGA, cannabichrominic acid, or CBCA, cannabidiolic acid, CBDA, and delta-9 tetrahydrocannabinolic acid, or THCA. And all of these acid compounds can undergo decarboxylation when heated to provide the neutral phytocannabinoids. And it's understood that the psychoactivity of cannabis is attributed to delta-9 THC, the neutral product from the decarboxylation of THCA. There are hundreds of other phytocannabinoids that are present in the plant, such as these homologs in the lower left-hand corner, CBDV and THCV, in which the 5-carbon alkyl chain has been substituted biosynthetically. It goes along a pathway which produces only a 3-carbon chain. So instead of starting all the way back in the plant from olivotolic acid, it starts from veranolic acid, a 3-carbon chain, and then it produces phytocannabinoids that are similar with that 3-carbon chain. Other phytocannabinoids are present in the plant, but they're from degradation pathways, such as isomerization or oxidation, that might occur during aging or handling of the plant, or the extraction process. And these include psychoactive compounds such as delta-8 THC and cannabinol, or CBN. It's really important to note that these products, such as delta-8 and delta-8 THC and cannabinol, are not produced biosynthetically in the cannabis plant. And so with that kind of biosynthetic pathway more well understood, we can talk more about cannabis in general. So starting in 1970, cannabis was designated as a Schedule I compound under the Controlled Substance Act, and the DEA was charged with overseeing that Controlled Substance Act when it was merged with several other government agencies in 1973. So the last 28 years is when we see a lot more movement on cannabis becoming available for customers or consumers. That was the introduction of California's medical marijuana law, or medical cannabis law in 1996. And then over the last 10 to 12 years with recreational cannabis starting to appear in Colorado and other states after that from 2012. But one of the more significant changes and complexities that arose from it was the 2018 Farm Bill, which legalized hemp production and products derived from hemp, such as CBD. It defined hemp as cannabis sativa L containing less than 0.3 dry weight % of delta-9 THC and set the procedure for testing using a post-decarboxylation or other similarly reliable methods. And any cannabis sativa L above that 0.3 dry weight % of delta-9 is still considered scheduled under the CSA and under the DEA's drug code of 7360, which defines it under the antiquated term of marijuana, where you're spelling that with a J or an H. And whether you call it hemp or marijuana, it's all still cannabis sativa L. It's just had testing that lumps it in to one of those two buckets. The DEA has other drug codes under the Controlled Substance Act that encompass products from cannabis that don't meet the definition of hemp. So cannabis extracts that are not from hemp are covered under 7350, and 7370 covers tetrahydrocannabinols. Both of those, both the THCs from cannabis and those that are synthetically produced. And so these issues start to arise from the Farm Bill and some of the different language used in it when people start to talk about delta-8 THC produced from hemp-derived products such as CBD. And so we've gotten to this point where we're starting to see new acronyms pop up and new products that are introduced that are said to be hemp-derived or hemp-compliant. And it's just adds a new layer of complexity that we have to deal with.
Got it. Chris, would you like to add anything to that before we move on to the next question?
Yeah, so I think that the Farm Bill, everybody quotes the Farm Bill to me all the time, saying that 0.3% delta-9 THC is what the Farm Bill says. It is really important for regulators to understand that there's two parts to that. The assessment of that level of THC is critical. The assessment that says that the measurement of delta-9 THC has to be via a post-decarboxylation method. There are multiple ways to do that, but in all cases, that means that you are taking into consideration the potential for any THCa that is in the flower to be converted to delta-9 THC. And that consideration means that your total THC is what's used for that assessment. So we see a lot of interstate commerce. We see a lot of retail operations that are selling what they call the THCa hemp. And essentially that's just marijuana. There's nothing different about it.
Right. Yes. The legal status of THCA is very confusing. THC is legally regulated. Legal status is up in the air for THCA. So what's going on here and more about why this is problematic?
It's challenging because many of the states have created regulatory pathways for production, distribution of marijuana products, first under medical programs and then under the recreational or adult use programs. Those programs are pretty tightly controlled in terms of what processing activities are permitted, the licensing, the oversight from the individual state regulatory board. Almost every state requires a significant amount of analytical testing to ensure that what's getting into the consumer's hands do not represent a health concern. And so you have now these products that are completely unregulated in many cases that are now competing directly against the regulated market. And so the producers of these unregulated products are not paying the same licensing fees. They're not subject to the same oversight. They're not subject to the same quality concerns that regulated production entails. And so When we look at some of the synthetic derivatives, we find them to be highly contaminated. We've tested thousands of these, and 100% of them have had contaminants from synthetic byproducts and other contaminants. So it is a significant health concern.
So on that, synthetic hemp product testing currently is overall flawed, completely complex. Let's try to break that down. I'm going to bring up this slide. All right, we are looking at Delta-8 THC gummies. What are we looking at here?
So we're using, like most laboratories, liquid chromatography for the analysis of most of our products. And under that analytical approach, we would typically see one peak for each chemical signal that is there. And if this was a truly Delta-8 product, you would expect to see one peak, maybe a little bit of CBN, which can also be found in the mixture. But typically when we look at these synthetic products, we will see somewhere between 7 to 30 contaminant signals. These signals are not observed in any natural products. So these are unnatural chemical compounds for which there's no toxicity information available. In the majority of the cases, there are not reference standards. There's not certified reference standards for us to be able to identify and accurately quantitate the contents of those. In addition, we also know that the analytical approach sometimes takes multiple different analyses. So in this particular example that we're showing, this was before we knew about these hiding peaks, but there's chemical contaminant signals that are literally hiding behind the Delta-8 THC signal. So that means any assessment of purity for these compounds is being overinflated. And so if you look at the chemical signal that we're showing, 47% of the recorded signal are chemicals not found in nature, again, with no toxicity information available for those. From a health perspective, that's incredibly concerning.
Got it. Absolutely. So given what we've already discussed, let's pause and ask ourselves, what are some things we would like regulators and lawmakers to understand about what we've discussed so far?
I think one of the things that we want people to understand is that there are products being sold as hemp compliant and somewhat considered safe just because they've got a C of A that looks similar to something that you would get from a dispensary, whether it's a recreational or a medicinal program. We want people to understand that some of these compounds that Chris has displayed in that chromatogram haven't been identified, and some of them are purely synthetic. Some of them should still be covered under the DEA's guidance, because while they might be found in very minute quantities in cannabis itself, they can't be, you can't expect somebody to have gram quantities of these materials without producing them in a synthetic setting. And that's where it becomes very concerning, because as Chris said, you don't have that level of testing to make sure that these products are safe. But people are turning around and saying, well, this is a hemp-derived product and it's totally safe. You can go and ingest it however you want.
I'm going to share this slide here on the decarboxylation.
Right.
Decarboxylation.
Yeah, I mean, this is what Chris and I were talking about with the THCA flower that's being sold. And these are being sold in marketplaces that don't have a recreational cannabis program. And they'll say, oh, it's THCA flower, but it's really important to understand that when you're calculating out the total amount of THC, you're taking into account the THCA there. That multiplier of 0.877 represents the difference in molecular weight between THCAA and delta-9. But that THCAA that's being sold in flour or gummies or whatever as hemp compliant is going to decarboxylate and provide the psychoactive delta-9 THC. So it's this, what they think is a loophole, but really is just them trying to get away with something. This slide demonstrates the complexity that we're dealing with. We put this slide together to try to show the multitude of compounds that you could expect. You can expect isomers where the double bond moves, or a structurally different isomer of THC. You can expect homologs where that alkyl chain changes from a 5-carbon chain to a 3 or a 7, which have been found in cannabis. But we're even seeing products out there with 8 carbons or more, which have not been scientifically proven to be in cannabis. So we have people who are synthetically producing these compounds and just saying, well, if 7 carbons produce a psychoactive effect, I'm sure 8 carbons will too. And then we have people producing analogs such as HHC or the THCOs, the acetates, or other compounds that haven't been tested to know their safety, their affinity or efficacy. And there's just a lot of unknowns about these. And a lot of them are still covered under the Controlled Substance Act as well, but they're being sold as hemp compliant or cannabis products.
Got it. So you mentioned that THC loophole. I've been hearing a bit about that. Can you dive a little bit more into what that means and why we should care?
So part of that loophole is just a misunderstanding or the lack of knowledge on the contents of the Farm Bill. So the total THC that includes the THCA as a component of that total THC assessment. So there's a lot of misunderstanding about that. The wording within the Farm Bill talks about isomers and derivatives being exempt from the Controlled Substance Act. And so we see a lot of these compounds like delta-8 would be a synthetic that you could argue is legal under the definition of the Farm Bill. HHC can also be made from CBD. But we see a lot of these other compounds that were in one of the last slides that were the THCP or THCjd, the 7 and 8 carbon homologs. These are being manufactured and produced under the Farm Bill as a Farm Bill compliant product. The only problem is these cannot be made from CBD. These are not able to be semi-synthesized from anything in hemp. So these would represent pure synthetic compounds. And as pure synthetic cannabinoids, these would be controlled substances under the DEA. But just because they look like some of the cannabinoids and producers are claiming that they're hemp-derived, they feel like they're free to manufacture these. And again, as Jeffrey was saying, there's some evidence that when you extend the chain length from the 5 carbon to 6 carbon, 7 carbon to 8, that binding to receptors gets much stronger. And so these are potentially significantly more psychoactive or more intoxicating than delta-9 THC itself.
Yeah, I mean, we've seen that for the THCPs, the original paper described delta-9 THCP as being like 28 times more, having a 28 times greater binding affinity. But that doesn't always equal efficacy. We do see reports on different forums and chats that say, sometimes refer to THCP compounds as the weekender, because you take it and it will knock you out for the entire weekend. But when a scientific paper that came out in 2021, I believe, they used our standards to quantify how much THCP was being seen in domestic cannabis, and they were finding minuscule amounts of THCP. So again, that gets back into the fact that, yes, some of these compounds might be there as minor phytocannabinoids, but they didn't come derived from CBD. You can't make THCP, the 7 carbon chain, from the 5 carbon CBD. And they're in such small amounts that I don't think that there's enough acreage out there to plant enough cannabis to get a kilogram of THCP. And I've seen that offered on different forums for sale. And so it becomes a little bit dangerous when we start to see these very psychoactive compounds or high binding affinity compounds being offered for sale as if they're the next best thing and obviously safe and hemp compliant when they probably are not, when they most certainly are not.
Mm. Great, thanks for that. So how all these laws are written can also be very confusing, especially when each state defines their own rather than looking to some singular federal law, some source of truth out there. What are some examples of states you've seen where the lines are very blurry and we certainly need further explanation and clarification? Chris?
Sure. So keep in mind that at the federal level, hemp was defined as at 0.3% as measured by that post-decarboxylation technique. That last caveat is critical. When the states started writing their own definitions for their own state hemp program, the majority of the states have copied that word for word for the most part. And so the state definitions of hemp mimic that directly. Again, there's still some confusion about what that post-decarboxylation method means. So there is still some ambiguity, even if within states that have that, as far as what is legal and not. There are a couple states where their definition, they have only copied the 0.3%. I think Mississippi is one of them where I could not find any further clarification on that post-decarboxylation measurement. And so that does lead to confusion. Is the state's definition of hemp supersede the federal definition? I don't know the answer to that question. There's states like, I believe, Maine. Maine does not include that post-decarboxylation method directly in the regulations that I could find, but in the guidance documents talk specifically about using the total THC for assessment. So even within the state's own set of documents, there's a little bit of ambiguity there. And so I think a lot of it is just the complexity that we're talking about in terms of the synthetic compounds, how these synthetic compounds are created, whether they're a simple isomerization or a CBD conversion, or if they're purely synthetic. Are these synthetic compounds that mimic naturally occurring ones like THCP? Are they synthetic compounds that are not found in nature like THCO? There's just so much complexity and so many different operating procedures to produce these that I think it's overwhelming for regulators and legislators at the state level to understand what's going on, and therefore they don't really know how to craft regulations or guidance to address all these individual pathways for creation of all these myriad of synthetic products.
Jeff, did you want to add anything to that before we move on?
No, I think Chris did a really good job of talking about the state level. You know, from the federal level, looking down, we've had a lot of back and forth with the DEA because we ask them for specific guidance on different phytocannabinoids. And we've often gotten responses which surprise us because they'll say, you know, of course it's still scheduled. Why wouldn't this be? And we've had them go back and say, no, we've decided, you know, something that we said wasn't scheduled is now scheduled after further clarification. But a lot of it depends on what is known in the literature. You know, is this compound known to exist in cannabis? And if it's known to exist in cannabis and has psychoactivity, then they probably are going to schedule. If it's not known to exist in cannabis, but it is known to have psychoactive effects, then they'll say, well, this is covered under 7370 because this is, you know, a THC. If it falls under the definition of a THC and it has that pharmacological data to support it being a psychoactive compound. So there is just levels of confusion at every single level with regard to this. And I think that, the states are the experimental labs that are setting up cannabis for changes in scheduling and maybe descheduling eventually. But it's going to continue to be a slow process as we're all figuring this out.
Yeah, thank you for that. So in my next question, David, I'd actually like you to jump in for this one as well. Hemp product testing is not currently required in the way that legally regulated cannabis industry products are. We know that. So let's talk about the requirements for legally regulated versus unregulated and the myriad of problems that can arise from that. David?
Sure. Thanks, Bethany. So, this is, as I think, Jeff and Chris have really well articulated, like a complex topic, even for myself with, science background. We're getting into really advanced chemistry and looking at the chromatograms and what the heck is in these products. And, the Farm Bill, think about the production, right? That, from my understanding, is based on like the farming and cultivation practices, the growing practices. But what happens after? Right? It's one thing to grow a plant in the ground, just like we grow corn, grow soy, etc. Where does it go from there? And does it go into an industrial process, or does it go into a consumer product? Something that, and when I say consumer product, I mean something that we actually consume. We ingest, we inhale, we put on our skin, we put in our soap, on our hair, etc. And that's where the game changes, right? This is about consumer safety. And when you go to a gas station and you know that the soda that you're buying is consistent, no matter whether you're in Massachusetts, Arizona, or anywhere in between, or even, internationally. And so what's missing here is those basic safety standards. And we have that in the adult use and medical use marketplaces where they're controlled under state boundaries. They vary, but generally based on 5 to 10 plus years of operations, we're not seeing attributions of adverse events related to products that are sold and regulated and tested in state marketplaces. We've got Canada that's had a full adult use marketplace since 2018, and they also can confirm that based on the data from a public health standpoint. So when you look back to when it leaves the farm, where does it go? That's where the gap in regulations are. And there's some real low-hanging fruit that I see that would really address a lot of consumer safety issues, because I've heard on the opposite side of where states have tried to push for regulating these products under their existing medical or adult use frameworks, and there's arguments for them being overregulated. But so what is that smart regulation from the consumer safety? Things like universal label standards, like what should be on that product to inform a consumer that it has 100 milligrams or 1,000 milligrams, and how do we address the complexity that THCP might have a 28x binding affinity than regular THC, and how does it interact with our body? These are like lock-in keys, right, for folks that maybe not be as familiar with how these binding affinities work. And you've got the left hand and the right hand, those are your different isomers, and they interact differently based on the lock and key process. So the ability to interact with these receptors in our brain that causes these alterations is critical for us to understand. The food industry and the pharmaceutical industry has to provide mountains of data to show that these products are safe, and we're not even there yet, right? So there's some really low-hanging fruit to tie into label standards. And then, number 2, production processes. What are these? You see, we've got 47% of that chromatogram that Chris showed that's unknown. Well, what is that? Is that just another cannabinoid, and is it a safe cannabinoid? How does it interact with the lock and key? Or is this something that actually could be toxic to somebody and actually put somebody in hospital or cause some sort of reaction that truly injures them? What are the production processes? What's the purity of those reagents that are allowed? Decaffeinated coffee is created similarly to how a lot of our cannabis products are, with CO2 supercritical extraction. That's a safe process. Pasteurization is a safe process. And in product categories, we don't even have basic product categories developed around this. We're just creating all these different things and not calling them anything consistently. So we really have to come together and create these minimum standards to ensure product safety. And this is where the lack is. Again, it leaves the farm. Now, what's the void at which we have major concerns in the marketplace around?
I want to add to that, a lot of the legal cases around the country that we see focus on the legality of Delta-8. All of them are ignoring the fact that Delta-8 doesn't exist in the commercial market as an isolated product. All the Delta-8 that we have seen, and I've been begging producers to send me one that's not contaminated, but they are all heavily contaminated with up to 30 different synthetic contaminants. And so these court cases are focusing on the legality of Delta-8 and not on the legality of these synthetic compounds which don't exist in nature with no known toxicity. And so that's a lot of my concern for these products.
Right. And I think the other thing is that David and Chris have kind of alluded to this. We're talking a lot about cannabinoids, but these Delta-8 products, or these products that are showing up in gas stations as hemp compliant, they don't have the same level of testing like bioburden and heavy metal testing. Literally, most of the suppliers, if you go to their website, they have the C of A showing cannabinoid content. And they just list, the usual 10 to 11, maybe they'll show more or less. And people are always trying to get the highest, the most active compounds in there. My biggest concern is that we're going to start to see people introducing other psychoactive compounds that hit the CB1 receptor and give that psychoactivity similar to THC, and we're going to get back into the synthetic cannabinoid craze from 10 to 15 years ago, where tin cans were being sprayed on plant material. And that's some place I don't think anybody wants to go to again, because it's going to damage the industry as a whole, and it's going to taint that ability to provide people with safe cannabis that they're seeking.
Awesome. Thanks for that. Continuing along, we are taking some deep dives into some chemistry, but also the impacts on the regulatory level here. So lawmakers and regulators and people like myself have limited understanding of testing. To reiterate the fact that some cannabinoids are natural, but others are synthetic. Let's explain this a little bit more for our audience and continue underscoring our concerns about this. Chris, I'm going to share this slide to support your response.
Yeah, so the actual production from CBD is actually pretty simple as far as dissolving the isolate in an organic solvent, refluxing it with some acid, and then neutralizing the acid, and then removing the residual solvent. The problem with that is that that conversion is not a singular pathway, not a singular synthetic pathway, but it's actually, there's probably 15 to 20 competing reactions. And so you can't actually get just a clean conversion of CBD to, say, Delta-8 THC. It amuses me when I see products in the market that include a pesticide screen as part of their safety profiling, because some people do actually test their products for safety. And I say, well, why are you testing it for pesticides? This in no way resembles an agricultural product. At the point at which this starts, and you're adding the solvents and acids, you're leaving the realm of an agricultural product and entering the realm of a purely synthetic organic product. And so to include an agricultural contaminant screening seems silly to me in the first place, but we do see that a lot, and that's part of the complexity of these compounds. When we test these, like I said, most laboratories use liquid chromatography. There are compounds that are not resolved or not apparent in liquid chromatography. As I said, they can hide behind different peaks. We call that coelution. We can use alternative methods like gas chromatography or supercritical fluid chromatography to get another look at that. And oftentimes we get better resolution of different contaminants that are there. But as I said previously, we've tested thousands of these products and to date have not seen one that is not contaminated. In the pharmaceutical realm, where these chemical syntheses are normal, they deal with a similar problem of synthetic byproducts. And under FDA and ICH guidelines, the producers have two options: either remove those from the product or isolate them and study them for safety to ensure that they don't represent, that their inclusion in a final consumer product does not represent harm to consumers. And so far, we do not see that happening. There are one or two producers who I see starting to do safety studies on some of their actual target compounds. But on these impurities, I have seen nobody doing that.
Jeff, would you like to add to that?
Sure. I think what some of these impurities that Chris is talking about, from a reference standard point, they're very difficult to even isolate, especially to isolate at the quantities to test. But there are some groups out there that are starting to look at these different byproducts and side products when they can isolate them and to start the testing. But it still depends on how the products were extracted. It depends on the process, as Chris just showed. He showed one simple setup, and that setup, the side products that can be generated depends on what solvent they're using, what acid they're using. And not everybody is using the same thing. Everybody's got their own recipe. And so he might get a chromatogram from one product of Delta 8, and it will show, 47% is not represented by the purity. But the next chromatogram he runs from a different supplier could be completely different. It could look very similar, but it could have a different profile and have new impurities that he's never seen before. As we're trying to provide reference standards, that makes it very difficult because often we're trying to play catch up. And we're trying to make these compounds, which are potentially unstable. They can be a very low yielding, and then they're hard to characterize because of that purification issue. You saw in the chromatogram that Chris showed how tight some of those compounds were. And you can try to imagine trying to isolate those and characterize those and then asking somebody like me, hey, can you make a reference standard like this? And sometimes we just have to say, we haven't figured it out yet. There are some that we have figured out and they've been very difficult to get to. But that's kind of the cat and mouse game that we play. Somebody makes something new. Chris looks at it, he identifies it. He comes to somebody like me and he says, can you make something so I can identify in this in my chromatogram again? And hopefully we say yes. I want to be able to say yes to Chris every single time, but sometimes I have to say not yet. So again, this is just a complex issue every step of the way.
Wow. David, would you like to jump in on this one before we move on?
Yeah, this is such, I think, an important point that ties back to, Chris, your reference of pesticides really just reminded me of the fact that, we went through this in the state marketplaces and we still have challenges with, 15, 29, 65 pesticides, et cetera. All that also had to, fortunately, these pests, fortunately or unfortunately, depending on how you look at it, these pesticides have been around for quite a while, and we have we have reference standards to be able to verify whether they're there or not. But now we're in a new world where we're in the pharmaceutical world essentially, and we're creating, these products, or these products being created, and labs like Chris that are expected to test them are getting to play Sherlock Holmes. They have very limited information. They don't know what chemicals were used, what processes were used. So to be able to even guess and go down the rabbit hole of what could this peak be is a lot of work. And then it also requires going another step upstream to Jeff and the Cayman Chemicals of the world to say, can you help me confirm what this molecule is? That's not a simple process. And in even the food world, this is a HACCP program essentially. You have to identify what are your risks, what could be in this product, and define those, identify them, and then show how you're mitigating or eliminating them, which depends on safety studies, as Chris mentioned, and then doing a verification step to, which is in some places, a product testing, a testing process or testing step to say these chemicals may or may not be there. And if they're there, they're at X levels. But when we don't know the ingredients, aka the solvents being used, when we don't even know what could possibly come out of that theoretical reaction, and then we don't have the, certified reference materials to test for them, we're running blind and we're not asking the right questions. So we really need to flip this on the script and say, we need more collaboration. If industry says, I'm using this process, I'm creating this product with these molecules, then they can work together with Chris and have a more educated decision around what should I test for? What method should I use? And that's how we get to a safe marketplace that addresses consumer safety. That's the void we really have to fill. That just putting out regulations does not address. But I'll stop. I could go on for a soapbox for a while there.
Got it. Yeah, I have a couple more questions I want to get to, and then also I want to try to make some time to answer some of the questions that came in from registration and the Q&A as well, and we'll try to fit all that in before the end of the hour. Next, we're going back to science class here. If a compound is plant-derived versus synthetically derived, how do we prove that scientifically? And I'm also curious what some interesting cannabinoids that are not found at high levels in the plant naturally. So we've got 3 slides. Again, going back to science class here. Jeff, if you would like to take us to class.
Sure, I'd love to. So when we're talking about plant-derived versus or synthetically derived, we can really look at this cannabidiol cyclization to delta-8 THC. We know, as Chris showed in a previous slide, that cannabidiol under acidic conditions can be cyclized to THC. And the major pathway is this one on the bottom where the phenolic oxygen adds into the isopropene group and gives us delta-9 THC initially, but under those acidic conditions with heating, rapidly converts to delta-8 THC. But one path that's less understood is the one on top, where the phenolic oxygen adds into the cyclohexene ring and provides delta-8 iso-THC, which also is prone to isomerization under these acidic heating conditions and can isomerize further to delta-4,8 iso-THC. These iso-THCs are not present in cannabis at all. So if you start to see them in your chromatogram, there's a great indication that those are going to be from plant-derived or synthetically manipulated cannabidiol conversion. So it's a fingerprint that says this THC is coming from this CBD conversion. And so we can look at that next slide. That's the chromatogram that I ran on these 5 compounds. Now, I have the benefit, whereas Chris is looking at some very complex things, we were just looking at these 5 compounds, CBD, delta-8, delta-9 THC, and the 2 iso-THCs. And you can see we get good separation of the CBD from the THCs, but you start to see coelution of delta-9 THC with the delta-4,8 iso-THC and the delta-8 THC with the delta-8 iso-THC. So there's not a really good separation for these by HPLC. And we had to go to GC, which is our next slide, to really see baseline resolution of these compounds. And when we were able to show this, it really opened up a lot of people's eyes, because it, again, is a fingerprint to show, hey, you can figure out if this is synthetically derived. Because what we're seeing now is a lot of labs are doing delta-9 THC production and calling it hemp-derived delta-9 THC, or they're just calling it delta-9 THC from cannabis, whatever they want to call it. But people are finding out, well, this is actually from a CBD conversion. They're just using better conditions to control that isomerization from cannabidiol to delta-9 THC and not continue on down the pathway to delta-8 THC. There are other ways to tell if something is plant-derived versus synthetically derived. The plant-derived CBD, for instance, when we've looked at the isolate that we've been provided, you can actually see the homologs that appear. So you can see the C3 homolog, the CBDV, you can see the C4 homolog, the CBDB, and you can see those in the HPLC, you can see them by LCMS. So there's ways to determine when these are synthetically provided. Another indicator that this synthetic marketplace is really taking off, when we talked about the longer chain phytocannabinoids and how they have a higher binding affinity and they're being sold as hemp compliant, we've seen the starting materials provided for those now being sold in kilogram quantities at much cheaper prices than they were 5 years ago. 5 years ago, if I tried to make THCP, I had to spend a lot of money to get the starting materials for that in my lab. If I go out and do it today, those starting materials are peanuts. And so people have realized, producers in both domestically and internationally have realized I can sell these starting materials because there's a great demand for them. And so they're producing them on larger quantities and that price has come down. So we understand that there is a synthetic marketplace out there for these synthetically derived phytocannabinoids.
Mm-hmm.
I just want to add one quick thing to that. When we talk about hemp-derived delta-9, I don't know what that means because there are two different pathways to get hemp-derived delta-9. As Jeffrey was saying, it is fairly easy to convert CBD into delta-8 or delta-9. And so some of the delta-9 that we see in the marketplace is a semi-synthetic product. We also see producers that are actually, they're doing an extraction and they are recovering the natural delta-9 residuals from their mother liquor and producing products from that. And so there are delta-9 products that are made, high quality delta-9 products that are made with natural delta-9. These types of samples do not have these synthetic contaminant peaks that we would see in a synthetic delta-9 product. But just having it on the label, I can't tell you what it is until we look at it analytically.
Mm-hmm. All right. So one more question before we take a couple minutes to try to answer some of these questions that came in through registration. Last but not least, let us underscore the risks to public health and safety in all of this, which is why we're all here.
So the FDA actually maintains a database of adverse events, and it is open to the public. I included the link there. But if you look at the instances of adverse events that were specifically tied to either delta-8 or delta-10, some of these isomers of THC, this does not include delta-9. This does not include spice and K2 and those other compounds which are considered synthetic cannabinoids. This data that I pulled was specifically related to the delta-8, delta-10, and those types of compounds. You can see that since 2018, the numbers have increased dramatically. I hear frequently that nobody's been harmed. There's one early case of a 4-year-old who died tragically from this, and I hear that single fatality being used as an excuse. There are actually 53 fatalities from these compounds. We as an analytical laboratory get contacted frequently, I would say multiple times a week, by parents whose children ended up in the emergency room, law enforcement where they found high school students that weren't breathing because they had been vaping delta-8 products. Delta-8 is not the evil culprit here. Delta-8 has some degree of safety that's been associated with it. It's the unregulated manufacture of these products that have unknown contaminants and synthetic byproducts and residual reagents that are creating a hazard for consumers.
Great, thank you for that. Jeff, would you like to add on to that before we move on?
I think there are, and Chris alluded to some health concerns. There are other health concerns that we might not know for years to come. The one that I'm thinking of is the acetates of THC or HHC. I've paper a few years back showed that the acetate moiety on the THC or HHC during heating and vaporization can turn into ketene, which is a known carcinogen because it will alkylate anything around it. And when you're putting that in your lungs and alkylating with ketene, that's potentially doing damage to your body that's not going to be evident in the next day or two. It's going to be evident years from now. And so those are, again, unknowns that we are introducing products where we don't know what the long-term health implications are. And we know at least cannabis has this millennia of basically a health safety record, and we know what the issues with it are, but we're now looking at compounds where we don't have that amount of data and assuming that it's good enough because it looks something like a compound that would come out of cannabis.
It's a multi-billion-dollar industry that probably, hopefully, might not kill you.
We hope.
And that's right. That's something that I'd like to think we've learned from history with whether it's been tobacco, sulfonamide, issues that led to the Pure Food and Drug Act of 1906 that we don't want to go down because we've now blurred the lines, right? Of cannabis is safe. It's been around for thousands of years. Well, what is cannabis? And we've incredibly blurred these lines in situations like the acetate, as you called out, Jeff, I think is key. This is the difference between having this molecule even in an edible or a gummy versus something that's being inhaled. And we're not asking those questions to date, and we're just lumping it all in together. The last thing I just want to say that I like to call out and remind folks of to bring it back is, just because it's Farm Bill compliant does not mean equate to safety. Just because it's natural does not mean it's safe as well. I've had poison oak on my body several times from backcountry hiking, and I love being out in nature, but I'm not going to go roll around in poison oak or poison ivy just because it's natural. So let's really take a hard look at what we're talking about here before we make these generic statements.
Great. Thank you all. We have 2 minutes left, and we've gotten some really great questions in through registration and the Q&A. Unfortunately, we won't be able to address all these, but one question came in. Do you feel, believe, or think that intoxicating cannabinoids, whether they're derived synthetic or otherwise, should be made available in the stream of commerce exclusively through adult use and or medical cannabis dispensaries? David, I think that you have a response for this.
Oh yes, hold on. Go back. So which question was that? Sorry, I was typing an answer.
Yeah, this one.
So, that's where I look at, one, it kind of doesn't matter what we believe, is what I'd say, because they're in commerce. That's, I think, as Chris mentioned, by some estimates that I've seen, a $20+ billion marketplace. So the cat's out of the bag. And so if we switch the conversation to what I think I've been kind of beating the drum here on, from my perspective, is if we look at a product safety and reducing harm, let's reduce the risk. There's no zero, but if we can start asking the questions of what is safe, and what level of data do we need in collecting that, then we can reduce. Let's go look back at that graph in 3 years from now that Chris shared about the FARS database and see those numbers start to decrease. That, to me, will be an indication of we are successfully allowing these products into the marketplace with sufficient controls. Until we see that, I don't know. Chris, I don't know, maybe you have got another thought?
So in my opinion, the pathway is clear. Legalization, just deschedule. Do not reschedule, deschedule, and a lot of this shenanigans goes away. A lot of this got to be where it is because of the prohibition in states where traditional cannabis products are not available, and so that's why people are trying to exploit these loopholes. So,
well said. Well said.
Right to your representatives and your legislators.
Excellent. Chris and Jeff and David, thank you so much. This has been an amazing webinar. Thanks to everyone who attended live. As a reminder, all past episodes can be found on the GMP Collective's website. I'll get that up soon in case you missed one. And be sure to subscribe to our channel on YouTube if you're interested in learning more about working with the GMP Collective. We offer strategic advising, subject matter expertise, data-driven solutions, and more, all designed to elevate your business's operations. Secure your success for your business now by visiting gmpcollective.com to schedule a consultation. Thank you for joining us. We'll see you next month.
Thank you.
Thank you, everybody.